So my discussion today is really focused on the opportunity that we have in this homecoming for personalized medicine to talk about where we are with respect to both the effectiveness of the work that has been done over the past 10 years by so many different individuals in different regulatory agencies in Europe, in the US, in Japan, as well as in the pharmaceutical industry, to be able to develop personalized healthcare. And my discussion today is aimed, it has multiple aims, and I think as you look through it, you'll see how these aims are sort of interwoven with each other. We have two speakers that follow this presentation that will discuss two different guidances that have come out recently at the EMA and at the FDA that talk about pharmacogenomics. And those two guidances have slightly different scopes, but they both converge in terms of the nuts and bolts of what it takes to do pharmacogenomics. I'm going to talk about today a smaller, more focused story. I'm going to discuss one that has to do with the difficulties that we have been able to work with regarding the design of clinical studies when you're trying to apply pharmacogenomics in rare diseases. And in the first slide, what I'm presenting is a paradigm which is really very worrisome. When people talk about rare diseases, they don't talk about personalized healthcare. And when people talk about personalized healthcare, they often do not talk about rare diseases. However, I would like to argue today that at least insofar as clinical study design is concerned, the problems, the challenges are exactly the same. They're not approximately the same. They are exactly the same. If you look at the issues having to do with clinical study design for rare diseases, we have a small number of patients. Personalized healthcare, also you have a small number of patients. You're talking about genetic markers for rare diseases and personalized healthcare. And you're talking about challenging clinical study design issues, both for safety and efficacy in both cases for rare diseases and personalized healthcare. The two terms are definitely not used interchangeably today. In practice, however, they will benefit in the future from certainly some regulatory guidances that may help both of these areas move forward. In some ways, when you think about rare diseases and personalized healthcare, there are big differences in terms of the urgency with which rare diseases and therapies for rare diseases have been moved forward, the advocacy of the patients that has been so urgent, has made it so urgent to have these therapies move forward. But when it comes to personalized healthcare, it's not clear that we have exactly the same kind of advocacy. We all think that there are major advantages, both in terms of patient care as well as in terms of public health policy in the implementation of personalized healthcare, and the previous speaker certainly laid out a very powerful example of how that can work. But the two approaches, the two types of therapeutic product development are definitely not on the same perspective from a regulatory standpoint today. Now, you come about realizing that these are the same sometimes by thinking through that in the case of a rare disease, first you have a clinical phenotype that describes the disease, and then you have some sort of molecular defect that is associated with that phenotype, and then you realize that there's a genetic marker that is associated with that molecular defect. In personalized healthcare, sometimes that intellectual vector is reversed. Sometimes you first think about a mutation that then leads to a defect that then perhaps, and you still have to prove that, leads to some sort of specific clinical phenotype. So it's important to keep in mind that we're still talking about the same problems, even though we have come about to think about them in very different ways. In my case, what I'm going to discuss today is the experience that Vertex has had in the development of therapies for cystic fibrosis. Cystic fibrosis is a multi-organ disease. You can find an impact across multiple organs in the body. We have as a goal in the cystic fibrosis program at Vertex to improve CFTR function and by doing so, reduce or halt disease progression. The strategy has been to develop an orally bioavailable small molecule modulator. And the idea is to combine this therapy with the current therapies that are available in the treatment of cystic fibrosis. The observation here is that you have a clinical phenotype for cystic fibrosis, and that clinical phenotype is due to a defect in the CFTR protein, and that defect is due to mutations that code for that CFTR protein, mutations in the gene that codes for the CFTR protein. So we are dealing with an approach where the goal is to be able to address the impact of those mutations on the function of the protein. The initial label for Kalydeco, for the drug that was approved both in the US and in Europe to treat cystic fibrosis, was made up of a number of studies, all focused on a single mutation, the G551D mutation, for patients that were 12 years of age and older. We also did studies for subjects aged 6 to 11. There was a safety study in subjects homozygote for the F508del mutation. And there was a fourth study, an open-label rollover extension trial for the subjects that completed STRIVE and ENVISION. The phase 3 study design was pretty straightforward, and this was a trial size that was focused on the detection of a 4.5% absolute change in FEV1. And the inclusion criteria included the G551D mutation, an age greater than 12 years, and a predicted FEV1 of 40 to 90%. And the results are summarized here. These have been published already. They showed a very strong change, a very strong effect, and a very, very big change in the absolute change percentage for the predicted FEV1. The impact on the health of these patients was immediate, and it was a very significant improvement in their ability to move on with their lives. The summary from this main study was that the primary endpoint was achieved, 10.6% absolute improvement. There was a 16.7% relative improvement in FEV1, lung function improvements throughout, and overall health improvement in weight gain and in other parameters. So that's the first mutation that is responsible for the molecular defect for which this therapy was tested. The question is, what do you do about all the other different ways in which this molecule may fail to work? And this is where the development program beyond G551D has been headed. If you look at the number of patients that have these different mutations, there's a group that has the gating mutations, and it's about 5% of the total patient population. 4% is G551D, 1% is all the others. There is a second group that is made up of residual CFTR function mutations, and I'll talk a little bit about those in a short while. And then there's everyone else. There's a large group where there is minimal CFTR function and where you have severe damage, and where the modulators are not expected to work very well. So as far as the modulators are concerned, we're focusing on this type of molecular defect associated with the gating mutations and the residual CFTR function. If you look in vitro at the defect itself, the molecular defect itself, this is what you see. If you look at the channel open probability through a patch clamp experiment, this is what you see for cells having the CFTR mutations that you see here. And as you can see in this type of experiment, it's a pretty clean result. You have virtually no channel. The probability of opening the channel is very, very low. If you look at the impact of the drug in this type of experiment, across the board, for all the mutations that are listed here, the improvement in channel open probability is very significant. And we would expect that all of these mutations would essentially behave as the original G551D mutation. So we are, in this case, essentially defining a class of patients. It's a class of patients, all of which pretty much have the same severe damage, the same severe molecular defect, and the same major response to this drug. Here's another group of patients. These are the residual patients. They represent about 10% of the total patient population. And in this case, there's quite a bit of variability in terms of the chloride transport. The efficiency of transport for this CFTR molecule is quite variable to begin with. And if you look at the impact of the drug on the efficiency of chloride transport, you can see that it's also quite variable. So in one case, we have a gating class that is pretty uniform, pretty homogeneous, and defined by a very important response to the drug. In this case, for the residual population, we have a heterogeneous population to start with in terms of the original damage that we're observing in the CFTR molecule, and quite a bit of a heterogeneous response as far as the drug response is concerned. And in summary, you can see all the mutations that would go into that first class where there's homogeneous response, the gating class, and the mutations that go into that second class. There are other characteristics that can help define a class when you're dealing with residual baseline transport. One of them is the fact that for most of these mutations, there is a coincidence of a lower incidence of pancreatic insufficiency. These patients are not as severely affected in terms of pancreatic function. If you look at the sweat chloride values and you look at where all of these different populations would be located, you can observe a range of sweat chloride results, and ranging from what a pancreatic insufficient patient would have all the way to what a normal individual would have. And in between those two, you will have the pancreatic sufficient patient population, and you will have CFTR-related diseases where there is a great deal of pancreatic insufficiency, and then the carriers and the normals. So this is the framework that we have to start from. These are the different kinds of molecular damage that we're working with. And the question is, how would you go ahead and design clinical studies to be able to test therapies for each of these types of damages? We have some conclusions so far. The gating mutations do form a homogeneous group, so they can be defined as a class and put into a clinical study as a class. We have the molecular phenotype for CFTR gating mutations, which can be used to define that class. So the in vitro data can be used to define that class. We have a shared molecular and clinical phenotype of the non-gating mutations. And all of these are associated with pancreatic sufficiency. So here's a way to obtain a patient class based on a clinical phenotype of pancreatic sufficiency. It's not all or nothing. That's what we're learning so far. In some cases, it's molecular phenotype. In some cases, it's a clinical phenotype that can be used to merge all these mutations into a single patient class that can then go into a clinical study. And what we're seeing here is, again, this interaction between genotypic evidence and phenotypic evidence. It isn't about getting a label eventually that lists 100 different mutations for which a drug will work and to what extent it will work. It's about getting a label that will be useful to patients and physicians. The conclusions from all the in vitro work that the CFTR gating mutations are a homogeneous group, so you can come up with a class of patients that could be used to run a clinical study for those mutations. That there are other mutations which are not as homogeneous as the gating mutations, and that in that case, you have to look at some other ways of defining a class. In this case, you're using a clinical phenotype to define the class, the pancreatic sufficiency phenotype. Keep in mind, not all mutations of each of these classes will have been known at any given point in time. I'm sure we will continue to find new gating mutations. I'm sure we will continue to find new residual mutations. However, you do get a label at some point in time. And the goal of being able to define patient classes is to be able to get a label that makes sense. It is not acceptable in personalized healthcare and in rare diseases to have labels that are made up of an encyclopedic number of mutations that just keeps growing in some sort of incremental fashion with no particular design and no particular logic. So these kinds of classifications are extremely important if we're going to succeed with the implementation of personalized healthcare. So we have a complementary bit of information between the genotypic and the phenotypic information. And these two types of information can be used to successfully come up with patient classes with which you can adequately power clinical studies. If you have a handful of patients that have one mutation, the wrong answer is to say, well, just make sure that you test all those 5 patients and see what happens. There has to be a better way, and it is very important that we be cognizant of this. So the development strategy for the CF patients that we're looking at within that group of 15% total of patients that are likely to respond to this drug is to first test if those CF patients with the other gating mutations benefit from Kalydeco. In other words, in this case, we have a gating class, run a study with the gating class. Test patients with R117H. This happens to be one mutation in the residual set for which there's enough patients so that you can actually run a study. And so we're running a study with R117H. Test if CF patients with evidence of residual exocrine pancreatic function benefit from Kalydeco. And that would be another study where you have now a clinical phenotype that is used to define the class of patients. And finally, and this is getting into something a little bit different, it's to pilot an N-of-1 strategy for use in patients with a less common or unknown CF mutation. So in this case, the idea is for all those other residual mutations for which there's just a handful of patients in each case, to be able to group them into one study, knowing full well that the response will be quite heterogeneous. That approach that is being proposed here is the use of an N-of-1 trial. In the case of the gating mutations, the study design that is being proposed is fairly conventional. It's just a case where you start with your two groups, placebo and Ivacaftor, and then you have a crossover after a 4-week washout, and you complete that with an open label period. It's a very straightforward study, and as long as you define your class of patients correctly, accurately, you can design a study which is actually straightforward to run. The case for R117H is even simpler because you have a single mutation, and in this case, you're just going to try to show the therapeutic benefit of the drug on those patients. The N-of-1 strategy is quite different because there, what you're doing is to try to have the patient be the baseline for its own benefit. So you start off with individuals, and you find what happens to each of those individuals in terms of therapeutic response. In this case, we have a pilot study that is planned where you're looking at the effect of the drug in CF patients with molecular or phenotypic evidence of residual CFTR function. So in this case, you approach the problem in a slightly different way. In this case, you assume there's essentially no class. You recruit from both the molecular phenotype as well as clinical phenotype classes, and you allow the study to absorb the enormous heterogeneity that you will be seeing in these classes. So that's the goal of the N-of-1 study. There are also enormous risks with this approach. We don't know yet what the outcome of this strategy will be, but it is certainly one that, in principle at least, ought to be able to absorb the heterogeneity of this patient population. So for CF patient populations in clinical studies, to summarize what you've seen so far, you have a big chunk of patients, 15% altogether, that are F508 del heterozygotes. Of those, there's a gating population, the population with a gating defect. These are patients that have a severe damage in their CFTR function, and these are patients, all of which respond to Kalydeco, whether they have G551D or other gating defects. Then you also have those that have the residual function that has already been mapped, and these are patients that also are pancreatic sufficient. So you have the choice of defining them on the basis of a group of mutations that we call residual, or on the basis of a clinical phenotype that we call pancreatic sufficient. So these are the populations that we're going after, and R117H is one of those mutations, of course. And these are the mutations for which the strategies that I just described are being applied. When it comes to molecular phenotype, are we talking about something new? No, it's been around for at least a decade. It has been defined both empirically, when you're talking about the concentration distribution of genomic, proteomic, or metabolomic molecules, the signatures, in other words, that have been identified over the past decade. There's also a functional or mechanistic definition, which largely refers to the kind of damage that we are talking about in the case of CFTR. This is what would define the gating mutations, biochemical or biophysical activity of a biomolecule determined by a genomic sequence affecting either its molecular function or its number of copies. And this is exactly what we're doing in the case of the CFTR gating mutations. There's a molecular phenotype which absorbs all the mutations within the class. Why molecular phenotypes? Okay, why are we talking about this? The therapeutic product development candidates often target specific patient populations, subpopulations, and they're characterized by individual patient genotypes. The populations are small, and you can use in vitro data to define what that molecular phenotype will be like, to be able to select patients for these clinical studies. Examples, well, you're going to be seeing a lot of those that have to do with CYPs, for example, where you have a whole bunch of different mutations that can cause a molecular defect, a detectable molecular defect in a CYP molecule. And of course, you may not worry too much about what the specific mutation was when you're going to run a clinical study. You will be running a clinical study for the molecular phenotype. What are some of the other examples that we can think about here? In Gaucher's disease, you have a rare disease where the molecular phenotype is a function of beta-glucocerebrosidase. The enzyme doesn't work. It doesn't work because of hundreds of different possible mutations that can cause the enzyme not working. And you're not going to ask a Gaucher's patient, hey, what is your genotype, before you give an enzyme replacement therapy. You don't do that. You don't do that because the development of that drug is based on your ability to be able to provide the molecular function, not on why there was one mutation or another mutation causing that damage in the first place. So no matter where the defect is in glucocerebrosidase, I'm sure it's extremely interesting to find out where that defect is. From the perspective of the therapy, you don't really care. You care that you're able to correct the defect. And the original mutation that led to that defect is still there, but you're not asking the patient what that mutation is. You're asking the patient, what is the class of mutations that you're in? There have been several rare disease product approvals over the past decade. In all cases, you're talking, in the case of the FDA, you're talking about small patient populations, large effects when you're talking about enzyme replacement. So in enzyme replacement therapies, to be fair, you can afford to come up with very small study population sizes in study designs, because you're going to expect to see a very significant clinical effect. In the case of Duchenne, the eteplirsen drug has been under development, and in this case, the approach is to skip exon 51 in the dystrophin gene. The molecular phenotype is the function of dystrophin. And the logic behind this particular therapy is to skip the insertion of an exon 51, into the final product, because that way you have a molecule that, while imperfect, can provide the patient with a healthier lifestyle than if they have the original major defect in a dystrophin. So that's the way it works. You're skipping an exon. Does it matter which mutations inside that exon actually cause the defect? The short answer to that is no. All you need to know is that there is a defective exon, and that there's a molecular phenotype defined around that exon, and that you're going to be skipping the insertion of that sequence. So no matter where it is, as long as it causes the defect, you're going to define your molecular phenotype. So to summarize then, the three kinds of molecular phenotypes that have been discussed here have to do with Gaucher and Duchenne, as well as with cystic fibrosis. And you can see that regardless of what the mutation is, you can group those mutations into classes that are associated with that molecular phenotype. This is very helpful when you're designing clinical studies. It's the only way, in fact, that you can successfully design those clinical studies. Okay, so these are close to the last few slides. In the cystic fibrosis workshop held at the EMA last month, there was a lot of discussion about non-pulmonary and other alternative endpoints. There was a lot of discussion about biomarkers in cystic fibrosis, but there was also a lot of discussion about novel trial designs that should be used to accommodate limited populations, increasing drug development pipeline, and the emergence of personalized medicine approaches. So the discussion that we're having today really is not new. It has already been brought up in trying to come up with ways to have some common sense when it comes to clinical study design. So the question, and this is the last slide in this presentation, is can we draft a guidance on best practices in the design of clinical studies for rare diseases and personalized healthcare? A guidance like this would have major impact across the board, both in terms of what is done with rare diseases as well as what is done with personalized healthcare. And the reason for that, as you may have seen today, is that the clinical study design issues are similar for both of them, that the molecular phenotype classifications that I've described certainly are tools that are common to both rare diseases as well as personalized healthcare, and that there is an urgent need to get some regulatory guidance in this sense. It's a case where initially you might think, gee, this sounds like a good idea, but there's a big gap between thinking that it's a good idea and having every single clinical division in every single regulatory agency agree with you. Thank you very much.